our next speaker, Anshul Kundaje, who will talk to us about, who's an assistant professor at Stanford, and his talk will focus on deep learning for genomic discovery.
Can you see my screen?
Yes.
And you can hear me, right?
Okay.
Yep. Awesome. All right. So firstly, thanks so much for the kind invitation. Yeah, I'm an assistant professor in genetics and computer science at Stanford, and my lab works on machine learning methods for understanding non-coding DNA. We primarily work on mammalian genomes, also to some extent in flies and yeast. But I'm very interested in actually, understanding regulation in plants as well. And so I'm excited and thankful for that I'm getting to speak here today, and hopefully this will open up opportunities for collaboration as well. Just to give some context, deep learning corresponds to a specific class of machine learning models. They're very powerful. They can learn representations from raw data. They've typically been thought of as very powerful black box predictors. Today, I want to show you that we can actually use these models to look under the hood, to understand what they're learning, and use them to actually make novel discoveries about the syntax and grammar of genomic sequences. As you know, there are large atlases generated nowadays, including, for example, plant cell atlas, there's human cell atlas, there's ENCODE, there's roadmap. These are the data sets we primarily work with, human and mouse data. You can think of all of this molecular profiling data sort of sitting on three axes. You've got the genome of some organism, in this case, human, 3 billion positions. You've got hundreds of different molecular readouts, histone marks, DNA-seq, RNA-seq, methylation, and you name it. And then on this axis is cell state. So this could be single cells, or it could be sorted cell types, or it could be tissues, or it could be data from multiple individuals or time points. So you end up with these massive terascale compendia of data. They are usually partially complete. Not every cell in this tensor is filled out. But this kind of data gives you a really great opportunity to apply machine learning models to do a variety of different interesting analyses. And I'm going to focus today very briefly on decoding regulatory DNA sequences and also using models to predict effects of non-coding functional variants and mutations. So I'm going to focus on the first part, which is trying to understand the syntax of regulatory DNA. And as you know, transcription factors are proteins that bind regulatory elements. They are sequence specific, so they typically bind. They have sort of landscapes of affinity for specific sequences, and they rarely function independently. They typically form complexes. They bind DNA in highly cooperative, competitive manners, often competing with or cooperating with nucleosomes. And so, one of the challenges has been to really, while there's been a lot of work to understand the intrinsic affinity, sequence affinity of these proteins, really understanding how evolution has encoded sequence patterns in the genome that allow for complex cooperative behavior of these proteins binding to DNA and how they regulate gene expression is still very difficult to figure out. Luckily you can use a variety of different assays nowadays to profile regulatory DNA. I'm going to show two here, like primarily ChIP-seq and ChIP-exo, which are used to profile protein DNA binding, and then ATAC-seq and DNA-seq, which are often used to profile chromatin accessibility. So given such an experiment, like in some cell type of interest, you could have thousands of these experiments or maybe just one. The question is, what can you do with it, right? So you want to understand how does DNA sequence drive this kind of beautiful protein DNA binding profiles or chromatin accessibility profiles. So you can model this as a machine learning problem, as a prediction task. And the way to think about it is the experiment that you perform that labels, that sort of has read coverage over the entire genome, essentially provides you a labeling of every single base in the genome, right? You get some quantitative score corresponding to read coverage from the experiment, or often this quantitative score is binarized. So you can, for example, you'll call peaks or no peaks regions of significant enrichment or not. And so what you end up with is you can take the whole genome, you can break it up into little chunks of, let's say, 100 base pairs each. And so you have millions and millions of these chunks across the entire stretch of the genome. And each of those chunks is associated with a label from the experiment, either binary or quantitative. And so what you end up with is a library of sequences, millions of them, tiled across the genome, each associated with a binary or continuous label. And your goal is to learn a model, a mathematical model that can map these sequences to transform sequences into kind of quantitative labels. And if you're able to do so successfully, you hope that this model would hopefully learn something about the properties of sequence syntax in the genome in these sequences that allow the model to make predictions about what the labels are, right? There's been a lot of work in this space, and it sort of followed this basic principle of taking short segments of DNA and mapping them to scalar quantitative values, either binary or like continuous values, right? But we recently came up with a new sort of class of models. We call these base pair resolution models, BPNet and Chrome BPNet. And what they do is just slightly tilt this on its head. Rather than summarizing the quantitative information from read coverage profiles in sort of windows, right? 100 base pair windows, you just count the number of reads. We decided to model the data at single base pair resolution. And why? Because the resolution of the assays has improved over time. You know, for example, ChIP-EXO gives you these beautiful base resolution footprints, as I'll show you in a few slides. DNA-seq and ATAC-seq at single base pair resolution can sort of profile high resolution footprints of transcription factors and so forth. And if you take this single base pair resolution data and you summarize it, or you coarse grain it into like 100 base pair chunks, 1000 base pair chunks, you lose a lot of the information that's present in the shape of these profiles that is in fact responding to the protein DNA contacts, right? So we really kind of model the data at single base pair resolution, the two strands. So our goal is to learn a translator. Think of it like a text-to-speech translator. The text is the sequence of the genome. The speech is the squiggly signal, read counts at single base pair resolution that you get from the assay. And we are going to fit a model, a neural network. A neural network is a fancy pattern detector. What it does is each of these neurons are actually pattern detectors, like motif detectors. You have multiple layers of pattern detectors. Each of them combinatorially build on patterns learned by lower layers. And so you can build arbitrarily complex models, which can learn extremely complex patterns, allowing you to map anything to anything, images to some labels, DNA sequences to some profiles. And you train these models conceptually very similarly to how you train a linear regression model. Each of the neurons are basically parameters, right? And you're trying to learn the parameters. You feed it millions of examples of sequences associated with labels, and the learning algorithm basically learns parameters that give you the best fit to the data. Now, one other interesting thing we've done in this work is, as you know, all of these assays have biases, there are enzyme biases and whatnot. And if you fit a very powerful model naively to some data, it is not only going to learn biology, but it's going to learn all the biases that exist in the data. You may have heard of similar problems occurring with deep learning models trained on other kinds of data, images and text, where they often learn all kinds of biases, some of which are not very pleasant. So bias correction is very important, and we have actually ways to input various kinds of bias tracks or learn biases de novo from the experiments themselves, regress out that bias and allow the neural network, a component of the neural network, to learn what we think is bias corrected sequence signatures, okay? So you fit this model, and what we do is we fit this model on a subset of chromosomes from the experiment, and we use it to predict on the remaining chromosomes. So in those chromosomes, the model has never seen the data. It can only see the sequence. You feed the sequence and you propagate the sequence with the model, and it makes a prediction of what the profile would look like. Now, when you do this, these are models fit to OCT4, SOX2, NANOG, and KLF4, 4 classic pluripotency factors in mouse embryonic stem cells. As we said, we fit on a subset of the genome, and we predict on a remaining subset. So you can really test what happens if you give the model new sequences that it has never seen before, and try to see what it makes predictions, what the predictions look like. So here I'm showing you 2 enhancers in the genome. And when you see OBS, that's the observed profiles, that's from the experiment. Red is predicted profiles from the model. Remember, the model has never seen these profiles. It has only seen similar profiles in other chromosomes. And now you just give it the sequence of this enhancer, and it makes predictions. There's one model for every transcription factor. There's one model trained for OCT4, for SOX2, for NANOG, and KLF4. And you can see the predictions are actually quite remarkable. They're very similar to the observed data. including little squiggles, the footprint widths and the fuzziness of the footprints, and so forth. So when we actually do this genome-wide or chromosome-wide on the held-out test chromosomes, and we compare it to replicate experiments, what we find is our models are able to predict profile shapes and read counts on par with the concordance between replicate experiments. So if you take one replicate and compare it to the other and see how similar they are, our BBNet model can make predictions just from sequence with accuracy that is on par with replicate concordance. So basically, it's really pulling out as much information as it possibly can from high quality data. So once you have this model, what's the big deal? You already have the data. Okay, so you can make some predictions. What's the point? Well, if the models have been able to learn these profiles this accurately, like if you take a sequence and translate it into base pair resolution footprints of transcription factors on sequences never seen before, it must have learned some biology in those parameters. So now your goal is to take the model and tear it apart and figure out what's going inside. So we have a suite of algorithms that we developed over many years that allow us to ask interesting questions to the model. We literally query it like an oracle, and the model tells us answers. And so it's really a question of building methods that can allow you to optimally answer specific types of questions. So one type of question you might ask is, okay, now I have a trained model. Here's the OCT4 enhancer, red distal enhancer, very famous. Here are the squiggly profiles, binding profiles of the 4 transcription factors. Tell me how the model is able to make these predictions so accurately. Tell me what is the contribution of every base in this sequence to this prediction. So what we do is we take the model, we can tilt it on its head, and we can propagate information backwards this time from the prediction to the sequence through the parameters of the model. And that allows us to give quantitative importance scores, contribution scores to every base in the sequence. When you add up the contribution scores, you get the profile. Okay, you get a contribution to the profile. So we can do this for every transcription factor, right? We have 4 different models, the same sequence, we have 4 different models. You can push the same sequence through the 4 different models. Each of those 4 models makes these predictions, and you can back propagate through the model to reverse engineer the contribution of every base. And you get these beautiful dynamic, sequence profiles. You can see some of these bases are completely nullified. The height of the base corresponds to the contribution. Some, you can see some of these bases really light up. Some of them are very context specific, like this one right here lights up only for nanog. This one lights up only for KLF4. And we can basically learn motifs this way as well. You can imagine you can learn like all the significant high contribution sequences. You can agglomerate them, learn motifs, and you can scan these profiles again. And you get these really beautiful context specific annotations of which bases are being used by different proteins to bind DNA. So you can see like different combinations of motifs driving binding of different proteins for the same enhancer sequence, right? You can do this for every sequence in the genome in any cell type of interest, for any assay of interest, as long as you have data for it and you can fit a model. What's exciting is you can do this not only for classical mappable enhancers, you can also do this for repetitive elements and retrotransposons, where it's very difficult to map short reads, right? So you can't actually map data reliably, but once you train a model on the mappable portion of the genome, you can easily make it predict on repetitive sequences like this one. These are various kinds of ERV elements. And you can see the model will highlight specific bases that are driving binding of different proteins to these retrotransposons. So we can actually interpret repetitive elements. And as you know, like a lot of retrotransposons have given rise to enhancers and promoters and regulatory elements. So we can start dissecting repetitive regions of the genome, even though we cannot map short read data to it through the predictive models. Now, what's more exciting is rather than just picking up individual features and looking at, these bases are really interesting, these motifs are potentially contributing, we can ask higher order questions, syntax. So one question we can ask is, how does spacing between any 2 motifs affect binding of 2 different proteins? So you have nanog and OCT4. We know that OCT4 binds this OX-OX motif. We know nanog binds this nanog motif. And what we can do is we can generate synthetic sequences in silico. We just take a backbone, some random sequence with similar GC content to real enhancers. We scramble it, so it's basically doesn't have any motifs that fire. And then we insert a nanog motif right here, and we insert an OX-OX motif right here, and then we systematically create constructs where we keep the nanog motif position fixed, and we move the OX-OX motif towards a nanog motif, right? And we push this library of sequences through the model. And now we simply animated the response of the model to, as you insert these various sequences, So you're seeing how the binding of nanog changes and how the binding of OCT4 changes as a function purely of spacing between OX-OX motif and nanog motif. And that's what's graphed here. There are two graphs. I mean, there are two plots. There's a red plot and a gold plot. The red plot is showing you the response of OCT4 to the spacing constraint. And the golden plot is showing you the response of nanog to this spacing constraint. And you see something really fascinating appear. What you see is OCT4 doesn't care about the spacing syntax at all. Its binding profile and strength stay pretty much intact. Classic pioneer factor. Nanog, on the other hand, responds very heavily. It is having the OCT4 has the OX-OX motif has a massive cooperative impact on nanog binding, and it is a function of spacing. As you can see, as the motif gets closer, you can see this kind of exponential amplification of binding nanog. But it's even more interesting is you see that as you get closer, you start seeing a periodic signal. This periodicity happens to be exactly 10.5 base pairs, which is a helical turn of DNA. So what this is telling us is that the model has learned a soft spacing syntax. It's not a hard syntax. It extends up to 150 base pairs. And then there's a preferential soft syntax where when the OX-OX motif and the nanog motif are on the same side of the helix, you get a massive cooperative effect in nanog binding, although OCT4 doesn't care, right? So you can actually infer directional causality, soft spacing syntax, things as subtle as helical periodicities completely de novo from the model, right? And this is through a synthetic sequence. We can repeat the same thing on genomic sequences. We can do an in silico CRISPR experiment. We can basically edit DNA. So here we take that OCT4 enhancer. And remember, we learned this OX-OX motif, and this little thing is a nanog motif. So this is a nanog profile and the the OCT4 profile and the nanog profile. Now what we'll do is we will in silico mutate these motifs. So we will knock out the OX-OX motif by scrambling it. And you can see now when you push the sequence in the model predicts complete destruction of OCT4 binding and a complete destruction of nanog binding. So even though we didn't touch a nanog motif, just destroying the OX-OX motif eliminates nanog binding completely. If you do the reverse and you break the nanog motif, you see that it has no impact on OX-OX. But in fact, it does have an impact. It attenuates nanog binding, but doesn't completely destroy it, right? And now we can do this for all the enhancers that have OCT4 and nanog motifs. And we can, again, average the plots as a function of distance to recreate this distance function. And you see something exactly like what we saw in the in silico synthetic experiment. Once again, you see asymmetric cooperativity as a function of distance, soft syntax with periodic preference, right? You can see it's fuzzier and weaker because you can't completely marginalize the effect of just these two motifs in actual genomic DNA. But in synthetic sequences, you can look at the precise marginal impact of just those two motifs together. We can then design actual CRISPR experiments this way. So Julia Zeitlinger, who's our collaborator, she actually took the model's predictions, as you can see right here. This is an actual enhancer. Here's a SOX2 motif. The model predicts this profile. This is what you get from the experiment. Very similar. Now what we do is we take the motif and we mutate it. The model tells us if you mutate the AG, you're going to get a pretty dramatic impact. You're going to affect the footprint right here, and you're going to attenuate the footprint pretty far away. I mean, you actually perform the experiment, that's what you see. You see a complete attenuation of the footprint right here, and a slight attenuation of the footprint right here. This is SOX2's cooperativity with itself, because it's the SOX2 profile that we're looking at. Now what we can do is the same mutation, but we can measure nanog binding. And once again, you can see the predictions of the model agree very nicely with the predictions with the actual experiment itself. We can now invert this, and there's actually a nanog motif sitting right here. So now we're going to mutate a different motif and look at, again, response of nanog to mutation of its own motif. And you can see a local effect. It's much more local. And now if you look at the mutation effect of nanog on SOX2, the model predicts no effect, and the experiment shows no effect. So the asymmetric cooperativity and the spacing syntax are beautifully validated by these high resolution CRISPR experiments. We can, of course, do this for every enhancer in the genome. It's very hard to do these CRISPR experiments at scale. Very quickly, I know I'm out of time, so I'll just quickly say that these models can be used to also predict other kinds of profiles, histone modifications, chromatin accessibility. They can be used to analyze single cell data. So here's a reprogramming time course from fibroblasts to iPSCs, and we can track specific loci, like here's a nanog locus, starting from fibroblasts all the way to iPSCs through pseudotime. And we can look at pseudobulk samples from these. We can fit models, and we can literally read out dynamic, utilization of various elements, sequence syntax. Here are two CTCF sites, bookending a TAD, a transient CTCF site appears right here. It creates a new TAD that appears only in iPSCs. Here's an enhancer that activates very early, regulating nanog, and you can really see a constellation of OXOX motifs, KLFO motifs turning on and off. There's all kinds of beautiful sequence dynamics that you can just pull out using models trained on this kind of data. So out of time, but you can use this also for interpreting genetic variation, as you might imagine. You can propagate variants, actual naturally occurring variants or de novo mutations or GWAS loci through these models, and the models can make beautiful predictions about how the profiles change as a function of alleles. You can interpret the effects of the variants, what motifs they disrupt, what syntax they affect. And using this, we've been able to fine map, GWAS loci and Alzheimer's disease using single cell ATAC-seq data from adult brain. We fit one model for every cell type. We've done similar things for congenital heart disease. So we can even take de novo mutations from rare disease patients. Here we've taken single cell ATAC-seq data from the fetal heart. We've learned cell types. We fit our models to them. And then we can take de novo mutations from patient genomes. We can propagate them through models trained on each of these cell types. And we can home in on predictions like this. Like here's an example of a de novo mutation that is predicted to disrupt the SOX motif inside an enhancer that links and regulates the JARED2 gene. And we've again designed CRISPR experiments in iPSC-derived endothelial cells, and it beautifully validates the effect of that enhancer. You can see a pretty nice, decent, a small effect size, but a significant effect size of that specific mutation on JARED2's expression. So with that, I'll just conclude and say that, we can actually use large-scale molecular profiling data to decipher genome function at the level of sequence. Neural networks can map DNA sequence to a whole variety of molecular profiles at unprecedented accuracy, but they are not completely uninterpretable black boxes. We have a series of interpretation tools that you can leverage to decipher functional nucleotides, motifs, higher order syntax. You can use them as simulation engines. You can interpret repeats. You can, of course, use them to predict and interpret functional genetic variation, and you can use them to target, specific targeted design of downstream validation experiments, which may not be feasible to perform always in high throughput. And with that, I'll just thank my lab. These are the folks who did the work. Thanks to Ziga, a summer student who really built the BPNet model a few years ago. And then none of this work is possible without the amazing collaborators and the funding sources.
Awesome. That was fantastic. Thank you so much. There's a question in the chat about whether you checked the model in Express Commission. If you can tell anything about like dynamic levels of transcription factors, dynamic binding.
Great question. So yeah, one clarification is important. So the models we currently build are sequence-only models, so they really can't make predictions out of context. So that means we primarily use them as an oracle for, it's like taking the experiment and shoving it into a model, and everything you want to do in the lab in that exact cell context, you could do in silico first, and then generate millions of predictions and mainly use it as a discovery engine, right? So it's predictive as far as you keep the context the same, right? We have other models that can go cross context, but a sequence only model cannot go cross context because sequence is the same in all contexts, right? Here, the primary goal is simply to learn a mapping of an experiment onto sequence and then interpret it and come up with all kinds of interesting biology and then guide downstream experimentation, right? So yes, if you have a time series, like a stress response, you can absolutely fit models to every time point, right? And like what I showed you for the fibroblast reprogramming time course, doesn't have to be single cell, can be bulk, whatever it is. And you can track how the same enhancer gets, differentially interpreted across time. You can do all kinds of things, right? So it's mostly an interpretation engine, and it's predictive as far as it goes in terms of perturbations that you can make to the sequence, holding context the same.
I had a somewhat related question,
yes.
Which is, do you know how much of the binding profiles is completely dependent on just sequence versus how much is influenced by other binding pathways?
Great question. So I think break that on. I'll break down a little bit more. So we model two properties of the profile. One is just the shape of the profile, and the other is the actual density of reads, which sort of captures occupancy. So we can model the shape of the profile literally perfectly. Like the local sequence can completely explain profile shape. That's why I said that basically you cannot distinguish between replicates and our model, right? But the actual occupancy, right, the total read counts are not completely predictable for two reasons. We still do really well. We're like in the 0.78 to 0.85 range of correlation, in peak regions, but it's not like 100%. It's not like replicate concordance. And the reason is because we are modeling currently local sequence. We're just capturing what like a 2 kb or 4 kb sequence contributes to binding at that location. But clearly we know there are long range enhancers and like, the genome is a graph. It's not, there's long range interactions and there's trans effects and there's, all kinds of other the trans environment has effects too. And that's something we can't model, but we have very nice evidence that even, that the density head that models the occupancy is really maximizing everything you can pull out from local sequence. I can talk about more at some other point how we know this, but we know this by comparing to in vitro experiments and comparing what we can do, how much we can pull out of the in vivo models comparing to in vitro models, and we can basically do extremely well. We can get extremely accurate affinity estimates from the in vivo models, even though they are trained on kind of noisy data with the sequence only context.
Cool. There's a question from Ken. Thinking about translation of these techniques to plants, what is the bare minimum amount of data or features needed for learning cooperative behavior?
Great question. So, one experiment is enough. Like if you have an ATAC-seq experiment in some cell type from some plant, and hopefully some proportion of the genome is mappable uniquely with short reads, you can absolutely fit this model, and you'll get pretty much the same kinds of information we can get. Of course, if you have multiple readouts, then you can really play this kind of cooperativity game, right? Asymmetric cooperativity. Those things usually require multiple readouts, but we can still pick up syntax. We can still pick up dependence between motifs. We can pick up, but we can't necessarily pick up directionality always, right? But we can fit this. So I'll tell you what kinds of assays the models work for currently. They work for ChIP-seq, TF, ChIP-seq, ChIP-exo, cut and run, cut and tag. They work for ATAC-seq, DNA-seq, single-cell ATAC-seq, ProSeq, ProCap, MPRA, StarSeq, pretty much everything. We've not yet built chromosome scale expression models, like, in vivo expression models. Those are coming next, hopefully by the end of this year, because those need to see the entire genome. They can't just work with a local sequence because of long range effects. But we'll get there. We are close. By the end of next year, we'll have in vivo expression models.
Cool. Another question, can we take one transcription factor ChIP-seq data set from one species and build a model to predict the targets in another species for the homologous?
Yes, we've actually done that. It works pretty well. We have a genome research paper where we do human mouse transfer, for example. It works pretty well, but we found a specific case where it breaks down. That's the beauty of having, like, interpreting the models. You can actually tell what's going wrong, like why the model's making mistakes. And it specifically makes mistakes in species-specific retrotransposons. So like the mouse retrotransposons have a very different origin than human, right? And so that is a domain shift. Like you've never seen any sequences like it, right, in training. And so when you make predictions, you get biased towards human-specific repeat architecture. But otherwise, the syntax and stuff is very similar. And one cool thing we've done, for example, is we've trained these kinds of models in single-cell ATAC-seq data in mouse models, where you can get very high-resolution time course data in a specific disease model. And then we can, even though we train the model on the mouse genome, we can just make predictions on human variants by shoving human sequences through the model. It works actually beautifully. It works very nicely. In fact, better than the human data, because in this case, we were looking at hearts, and it was very hard to get high resolution time course of atherosclerosis in the humans, but we were able to get it in the mouse. And there's a specific cell state, where these variants really erupt, and you can see it through the mouse models better than the human models.
I think we have time for just one more question. from Josh Cohn, does peak size affect models broad versus narrow peaks?
Yes, absolutely it does. And that's one of the reasons we moved away from the whole peak-like model, because as you know, peak calls are pretty arbitrary. You know, like there's a peak caller, you run 3 peak callers, they'll give you different segments, they'll call more peaks or less peaks. We got tired of kind of modeling peaks. And so we just were like, well, let's scrap peaks and just model the data at its absolute raw resolution. So we literally just take BAM files, we count number of 5' ends at each position in the genome, and that's it. We just model the whole genome. And it's liberating, firstly, because you don't have to fudge around with peak callers, but more importantly, you're leveraging the maximal resolution of the data. So footprints and all of this beautiful, signal at single base per resolution, you can suddenly start picking it up, which you are losing with like peak-based methods. So we adapt to different assays, like histonemarks typically require broader receptive fields. So we model like 70 kb, 100 kb sequences. For transcription factors, chromatin accessibility, we can get away with like 2 kb context. So you expand or contract the model, but otherwise we don't care about peak calls.
Cool, thank you.